Good afternoon, everyone. My name's Phil Bourne. I'm the Dean of the School of Data Science. I haven't been saying that for very long, so it's sort of kind of neat. This is one of a series of what we call Fireside Chats, which seems a bit ironic when the weather's like 90-odd degrees. But we have these periodically, and the idea is really to have an informal discussion with a thought leader in various areas that relate to data science. So it's a great pleasure to be doing that today with Eric Green, who's the director of the National Institute for Human Genome Research. Thank you. NHGRI. Goodness, I've been away too long already. So Eric was, I'm going to ask him to sort of give a bit of his background and say a little about the Institute in a minute before we get into some deeper topics. But I just want to say how grateful I am to Eric. The 3 years I spent at NIH, he was responsible for what was called the Big Data to Knowledge program, which we'll talk about a little, I'm sure, when I actually got there, and proved to be a wonderful mentor and friend in navigating the whole of NIH. So for that, and the program itself. So for that, I'm very grateful. And it's a pleasure to have you here today to talk about various aspects around the general notion of the role of genomics in healthcare and how data science can play a role. But I'm sure we'll drift into other things as well. The idea is for us to have a bit of a back and forward discussion. If you do have a burning question or comment during our back and forward, then please make it. Otherwise, we'll reserve almost half the time in this hour to actually have questions that you can ask.
And maybe, Phil, I can make one request. I don't know people's backgrounds, and especially how many are biological backgrounds or how much people know genetics, genomics. So if there's a burning, I'm not going to get too technical. But on the other hand, if there's a burning question to help you contextualize what we're talking about when it pertains to genomics, just raise your hand. I'd rather answer it before I get too deep in than to have you wait till the end.
I mean, looking around the audience, many of whom I know, I'd say it's a mixture of people who are doing certainly data-oriented biomedical research. If I had to put a phrase on it, and that includes some aspects of data science. In fact, we could even have a discussion of what data science is in this context, which is always something that's fun and comes up. But perhaps you could start off just by telling us a little about yourself, your background, and the history of the Institute, and in fact, where data science perhaps stood in the Institute near the beginning.
Yeah, well, I don't even think the phrase existed at the beginning. But let me give a background both of myself and the Institute, which in many ways for the last quarter century have been interwoven. So I was trained as an MD-PhD student and chose to do pathology as my clinical specialty. And a key point is I graduated with my MD-PhD degree in 1987. And those historians in the room may appreciate that 1987 was the year the word genomics was coined. In fact, I can guarantee you will not find the word genomics in the scientific or medical press prior to 1987 because the word didn't exist. And so that was sort of prophetic in some ways because, well, as a graduate student, I did not work in anything related to genetics or molecular biology or any DNA-oriented science. But as part of my pathology training starting in 1987, after a year, I had an opportunity to go work almost full-time as a postdoctoral fellow. And I was at Washington University, where I'd actually trained as an MD-PhD student and had the opportunity to jump into a laboratory that was doing cutting-edge DNA analysis that very much was the beginning of the field of genomics. And I jumped in. And WashU at Washington University in St. Louis at that time was very much an epicenter for genomics just because it had several, just actually multiple very good people that were well poised to not only participate in this emerging field, but in particular, and the reason the field was named in 1987 was because there was this discussion of being comprehensive in trying to study all of the DNA in humans, for example, the human genome. And of course, this was exemplified by the International Human Genome Project. So 1987, the Genome Project was first being discussed, a field gets named. By 1987 or 1988, I was a postdoctoral fellow in a genomics lab, the right place, the right time, we were one of the first funded centers. We went in the first round to be one of the first US genome centers to participate in the Human Genome Project. And I got to help write the grant and help work on this literally from day one of the Genome Project, which is October 1 of 1990, for those of you keeping track. That's when the Genome Project started. I was there on the front line as a postdoc. So fast forward, I became a faculty member shortly thereafter. But you fast forward a little bit more, by about 1993, there was major changes going on at what was then the National Center for Human Genome Research. This was the part of the NIH that was created by the US Congress to lead the US's effort in the Human Genome Project. We got one of the first grants when I was at WashU. But the first director of it was Jim Watson. Some of you may remember that. By about 1992, Jim was no longer at the head of that center, and they were looking for a new director. And ultimately, by 1993, they recruited this guy called Francis Collins, a UVA alum, by the way, to leave Michigan and come to the NIH to lead the National Center for Human Genome Research. Francis said he was willing to do it after he was asked about 6 times, but he finally agreed to it. But one of the stipulations that he wanted to create not just the extramural funding part of the Institute supporting the Human Genome Project, he wanted to create on-campus governmental, what we refer to as an intramural component of the center, which means recruiting scientists to come lead their research programs at NIH. And he called me. I was one of the initial set of scientists that agreed to come, and I left WashU. It took me a year till I could finally get there, but by 1994, now 25 years ago, I joined actually as a mid-tenure track investigator, working at the Institute, working on the Human Genome Project. Finished that work, completed our efforts in the Genome Project, was involved in the Genome Project from beginning to end. By that time, I had taken on other responsibilities at the Institute, including leadership responsibilities. I was eventually the director of the intramural program, at what was then later became the National Human Genome Research Institute. We went from center to institute status. So one of the 27 institutes and centers at the NIH was the intramural director. And then when Francis Collins was eventually appointed to be the NIH director by President Obama, he then had to fill the vacancy that he left behind as director of NHGRI, and I applied for that job and was eventually appointed nearly 10 years ago. December 1 will be my 10th year anniversary as director of the Institute. So that's my history. I think the other key thing, which I know we're going to get into, is think about it. The Institute was originally created first as a center with a singular goal by the US Congress, lead the US's effort in the Human Genome Project. Genome Project ended 16 years ago. As you might imagine, we didn't want to go away. Of course, in many ways, the end of the Genome Project was just the beginning of everything. So of course, there was lots of exciting things to do, but the Institute has continually morphed over time. 16 years ago was a key pivot for us because obviously we had to completely redefine what we were about as we transitioned from the goals of the Human Genome Project to everything that has followed in the last 16 years. And we continue to morph and morph and morph, and that's what we'll get into that.
Yeah, let's get to that. But I just want to sort of bring where, at least from my perspective, and you can agree or disagree and perhaps say more, that sort of data science began to feed into this, because I find it, everybody sort of said, not everybody, a lot of people say, well, data science is something we've been doing for years, and many years. And you could say, all right, well, in the context of biology and biomedicine, it's really the notion that it started with bioinformatics, which then sort of grew into computational biology, that then sort of morphed partly into systems biology, and now it's become sort of biomedical data science. And my recollection of it being involved at Columbia University in the Genome Project, where we were, as you well know, there was a series of grants were awarded to work on different chromosomes.
Correct.
And we did the physical and genetic mapping of chromosome 13. The fundamental thing, from my point of view, was that we were not allowed to have money unless we had some computer person who could actually manage and actually analyze and help analyze data. And that at that time, Columbia didn't really have anybody at the medical school where I was, so I was the guy running the computer center. So that's how I got into it. And that, for me, defined the future. And so that, to me, that's kind of a little slice where data started to come from experiment and other cases. And now what we see today all around the grounds here is exactly that thing happening, but in every single imaginable field, from religious studies to history to politics to whatever. And so it's kind of like history repeating itself in a very large way. I don't know if that's your recollection of how...
Well, I think the wording, the progression of phraseology, I think is sort of my memory as well. So I think that these words just keep sort of evolving. And certainly the sophistication level of the challenges keep evolving. And I also vividly remember some of the problems some of the earliest centers had because they didn't have a computation person. You know, not even though that wasn't even well defined. I actually want to, just for fun, take people back, because I just think there's, when you think about where we've come, whether it's data science or computational genomics or bioinformatics, if you use any of the phrases you want, let's just go back to when the Genome Project started, 1990. Because when I tell people the following three things, they sometimes look at me like I have two heads. They can't believe it. That we would do something as outrageously audacious as the Human Genome Project when the state of affairs computationally in 1990 was as horrifically prehistoric as it was. And so the three examples I give, first of all, there was essentially, and again, I'm going to have to look around there. Some of you will not know what I'm talking about because you're way too young, and some of you will know exactly what I'm talking about because you're of a sufficient age. I mean, there was really there was barely a functional internet in 1990. I mean, when the Genome Project started, I just know I didn't even have an email account. None of us had email accounts when we were applying for the grants. I mean, so email sort of came on the scene. PCs didn't exist back essentially. So just think about the people who planned the Human Genome Project were doing so in the absence of email and a functional internet. That's a pretty crazy thought. So that's one thing. Then the second thing that I always remember back to the early days of the Human Genome Project was when GenBank would send around their entire contents on CD-ROMs. So there was first it was 2, and then it got to be...
It was actually tapes before it was CD-ROMs.
So I got them as CDs. You could sign up and they'd mail to you. There'd be 2 CDs and then 3, and then eventually it got a little bit big. It got to be a thick plastic box of 5. But just the notion of physically mailing the content of GenBank is just, it's like, I mean, did they deliver it on dinosaurs? I mean, you just can hardly imagine it. But then I'll tell you the third story I always like to tell, because I find it so overwhelmingly embarrassing that you would think there was just absolutely no way that the Human Genome Project could have been successful. Because I was so take you back literally the first 6 months of the Human Genome Project. You guys were working on chromosome 13. I was working on chromosome 7. Even the fact that we divided up the human genome by chromosomes actually makes very little sense in today's light, but it was just the way we would do it. But it gets so much worse than that. Is that my contribution, in particular, you could go back and read the papers, my contribution what we were really taking for a serious test ride on chromosome 7 was this idea of using the polymerase chain reaction, I hope people know what that is, PCR, as a way to detect landmarks across the human genome. And so all we needed was to create a tool that would allow us to then develop maps and eventually get things poised for sequencing. But this was early days. So all we were trying to do in my case was to develop these PCR assays that would detect an anonymous sequence just picked at random from chromosome 7. And it turned out that we didn't really have we personally didn't have a very good way of uniquely getting snippets of sequence from chromosome 7. But our collaborator did, who worked at the Department of Energy, a guy named Bob Moises. He was in the Department of Energy labs. And they had a technique called flow cytometry, which could be used to separate the 24 human chromosomes, and you could get a test tube that was mostly chromosome 7. So he was able to do that. And then he would clone that bits of DNA, and then he would into little pieces, and then he would get short 200, 300, 400 base reads of an anonymous sequence from chromosome 7. But here, the way he was doing this, by the way, again, this was he was doing this using radioactive sequencing methods where you literally look at X-ray film and you read the DNA ladders. Again, not even done probably for the last 20 years, but it actually gets so much worse than that. Again, the state of data science in 1990. He would physically read off the sequence and write it down in handwriting. And the way he would get me the sequence was to fax it to me. There was no email, so he would fax it to me. Oh, and by the way, then what you do, you have this stretch of 200 to 300 bases of sequence. You need to design two places in that sequence where you could synthesize an oligonucleotide. I may be getting geeky if you don't know what PCR is, but basically you just have to pick two stretches of about 20 bases in a fashion that you think this will work in this technique called polymerase chain reaction. Nowadays, of course, there's computer programs to do it. There didn't exist one then. So I used to look at this fax, crappy fax, by the way. It wasn't even laser printer paper. It was back then when it was fax rolls. It was that crappy thermo paper. I would stare at it and sort of guess what might make a good set of PCR primers, would manually do that and then go off and that was the state of affairs. I mean, when you think about the way we do things now, you would never do any of it then. The crazy part is actually it worked. I mean, and actually we were way ahead. Our map of chromosomes early on was one of the best ones, and it worked, but slowly but surely we figured out how to email sequence, we figured out how to, we wrote a computer program to design the primers, and so forth. But just imagine those three things when the Genome Project, how on earth that project ever was successful is beyond me.
When I tell this sort of thing to my kids or even classes, it's like people's eyes glass. It's like, okay, Grandpa, shut up. Let's move to the future. So we're going to do that in a second. Yes. So let me just add one thing that struck me in that era was that it was the first time I actually, me personally, which is perhaps why I'm sitting here talking to you, is that I saw a future. And it was about this notion of synergy between experiment and computation. In that as we were doing hybridization and we were doing assembly, and as we were doing that, we were actually coming up with algorithms to try and assemble these components. And that actually drove how we did experiments to actually, so it was this virtuous cycle which I thought was really cool. And I think that some of that, we see that obviously everywhere now. But it was, at the time, it was actually very new, even though the computing was really clunky. So fast forwarding to the 16 years, I guess it's a bit more than that now since the completion of the genome. Yeah. I mean, what do you think have been the milestones from NHGRI's perspective, and then projecting out from your point of view, and we'll dig into this a bit more, but where are we headed?
Yeah, so I think back on the last 16 years, what have been the highlights? And I would, what I would say first and foremost, which has been completely transformative and actually has created all the data science challenges, is clearly the improvement in technologies for reading out DNA sequence. So for those who don't sort of follow this carefully, that first human genome sequence produced by the Human Genome Project cost something like $1 billion, something like that. Very good investment, but it was about $1 billion, and clearly we never were going to accomplish what we wanted to accomplish in genomics with just one sequence, and at $1 billion we weren't going to have very many of them. So it was very key, and we embraced, among the many things we embraced 16 years ago, our responsibility as a funding agency to try to stimulate technology innovation in DNA sequencing methods. The good news is we stimulated it, but also the private sector got involved. And now we've basically created a situation, we've basically a millionfold reduction in the cost of sequencing a human genome. So now you pretty much can sequence a human genome for about $1,000, and you can quibble exactly on quality and whether it's $1,600 or $600, $1,000, $1,200, whatever. It's about $1,000. And what that has done, I mean, it's been transformative for everything, because now it means that sequence, it's not an international project, 13-year project to get a sequence. It's one day or a few days, and it also means that it enables DNA sequencing to be sort of a regular tool that can be used for all sorts of things, even beyond just generating human genome sequences, but as an assay tool in a laboratory. We've gone then from having one sequence of human genome to now we have hundreds of thousands of human genome sequenced. We have now amassed massive catalogs of how we differ in our genomes. We differ about 1 out of 1,000 places between any two people. That turns out to be, almost probably well over 100 million places in the genome are commonly variable amongst people. And we know where those are. We know what the variants. I mean, there's just this huge surge of publicly available data about human genomic variation. That's high on the highlight reel. And then, of course, we slowly but surely are figuring out all the complexities of function embedded in those 3 billion letters of the human genome. And we're only 16 years in of interpreting the genome. I think we have a long way to go. We're doing really well in the parts of the human genome that code for proteins, protein coding genes. We know there's about 20,000. We know most of them are. A lot of other complexities associated with them because they could, they produce more than one protein product in many cases. but we're getting it. But we've also learned there's a lot more complexity out there in the non-coding portions of the genome, a lot of efforts to sort of understand that. So that's all sort of at a very basic set of accomplishments: technologies, variation, genome function. But the other big growth area, I sort of put in sort of translational. I mean, this idea of what role does variation in the genome play in human health and disease, and in particular human disease. And there's just been remarkable accomplishments in cancer, remarkable accomplishments in rare genetic diseases, which are typically caused by mutations in a single gene, so they're simpler to unravel. And we're making inroads in understanding the bulk of human disease, and the bulk of human disease are more common but genetically complex diseases that involve more than one gene. And those are really hard, but there's been massive inroads, again, heavily catalyzed by better technologies so you can generate lots of data. And then the last highlight, before I get to data science, the last highlight, which is something I've particularly been pushing as director over the last 10 years, is clinical applications of genomics. So even moving beyond just basic knowledge of the role that genomics plays in human disease, but then operationalizing it of how can genomic information about individualized patients be used to tailor their care to make medical practice more precise. And here, lots more to be accomplished in the coming 10, 20, 30 years, but there's real success stories. I mean, it really is changing the practice of medicine in certain areas, and that's all very exciting. But I do think cutting across all of this has been massive opportunities because of massive challenges in data science. I mean, genomics absolutely is, I'm going to get to this more, but I mean, is I think the poster child for, at least in biomedicine, for the challenges of data science, because we're such a data intensive field and it's because our technologies drive it to generate data at just audacious scale.
So sort of moving into that data science.
We have a question? You want to
Yes, I mean, you are absolutely right that producing the data is very cheap.
Yeah.
It's $1,000.
Yeah.
But this data has to be transformed into information.
Right.
And information has to be transformed into knowledge.
Yep.
And I think that in these steps, we are not doing as much progress as we should. We more concentrate on producing new data than on use this data to something.
Yeah, well, I agree with you.
Well, actually, it's interesting because that was exactly where I was going to segue by saying, you were responsible for the Big Data to Knowledge project and the development of data science at NIH, and I got there, and perhaps you want to say a little about where it's going in the future. But just to put it in the context of BD2K, what I found when I got to NIH, there was much more concern about the BD than there was 2K. So I don't think we got past the notion of what are we going to do with all this data. You could argue whether we should be collecting it or not, but we already had a lot, and it wasn't necessarily usable. So, this
Some
is.
people did.
Well, some people did. I'm sure you'll get into that in a second. But I think that is, and so when we think about data science here, what I would call that BD part, which is, let's just call it data acquisition and engineering, is only part of what is really data science is about. And we're now starting to see really some major breakthroughs in the 2K part, whether it coming from various aspects of machine learning, natural language processing, all these kinds of things. So, I mean, perhaps from your perspective in BD2K and where the NIH is going, you could sort of carry us along that projection as to, from your perspective, so as we think about, partly of course it has to do as people out here start to write grants, what they should be thinking about, but that's putting it rather bluntly. But moving along in that direction.
No, I think that it could be a criticism, or at least it could be an observation. I mean, I think it's absolutely a fair point that the knowledge gain is probably lagging, and the big data challenges tend to occupy a lot of attention. And I know we were going to talk about that. I was trying to give that some thought because I think it is true. No, I think some of it is because the knowledge part's harder, and so that's always going to. But I was trying to think, why is it that I do feel a lot of what NIH's efforts in trying to get its act together over the last 10 years. Why is it that so much of that has been tied up in trying to deal with the big data aspects? And maybe some of it ends up being almost policy driven in some ways for the following reason. Is, one of the things that we're dealing with is an outgrowth of the Genome Project. One of the if you sort of say what are the 10 big contributions of the Genome Project, and I've actually written an article about this, one of the 10 big things coming out of the Genome Project, besides all the obvious other things, is this cultural change around data sharing. I mean, the Genome Project was really the first biomedical research project to share data widely in real time before publication, blah, blah, blah. And that sort of got baked into the culture of genomics and is now also getting baked in more broadly across NIH, but across other areas. But let's just focus on genomics. So what has happened, starting with genomics, especially as genomics gone beyond the Genome Project, was a very significant commitment on behalf of NIH to share genomic data as part of almost all grants. And you sort of step back and say, that's a great idea. We should share data. It was so pivotal for the Genome Project. Everybody's boat would rise if all data is shared. What, of course, that does is it gives you immediate challenge of, well, that sounds good as a value, but how do you actually execute on that? And how do we deal with this massive amount that just keeps going bigger and bigger and bigger? And then I think about, as we started to move into translational work in genomics, all of a sudden you were confronted not with just sort of dealing with that first human genome sequence, and even then we struggled with identity issues and privacy issues. That's a whole other story. But dealing with the fact that you're dealing with human subjects. And that brings some additional complexities. But you say we want to protect people and participants in biomedical. Absolutely, nobody would argue with that as a value. So now we have shared data as a value, accept it. Protect human participants in biomedical research, absolutely, value, protect it. You put those together, now you have some big data challenges. Who stores the data? How is it stored? How is it shared? And then, and now it gets expensive. Now then introducing cloud computing, introducing all sorts. So I think we're still struggling at the front end of just maintaining these values and executing them that's the reason I think it's been approximately heavy.
I mean, going beyond sort of the raw data, I mean, one of the aspects that we've talked about for a long time is, when I say we, I mean researchers and the NIH, is this notion of multi-scale modeling. And I think it's been a lot of talk without much success, but I think we're finally beginning to see fruits of that sort of labor. Things that, for example, relate to going in and looking at, now we have a reasonable, I won't say it's great, but we have a reasonable electronic health record where people can get access to lots of phenotypic information. Some of it's classified by ICD-9 and 10 codes and so forth. That's at one end, and then as you start to work backwards, you actually have information about, for example, drugs that people are taking, and then you can then test some of those propositions that you find in those situations in animal models, which brings you down a level. And that actually relates to, of course, sequencing that's non-human, which you've also been involved in a lot at the Institute. And then going further back into the genetic underpinnings of why these things are happening, and why that is in the terms of the biochemistry and the pathways and everything else that's involved. So we're starting to see the dots being connected, I think. And is that something that your institute is directly involved? I mean, how does that reconcile across institutes, and how does that reconcile with you? And what are, so there's a lot of, and then the sort of the data science aspects of that, how do you see that being handled?
So I don't think there's a crisp answer to any of those questions. I mean, here's the first thing I'd say. There's no question almost everything you just talked about we're interested in as an institute at some level. I mean, even if you look at, we began almost 15 years ago, recognizing that the marriage of electronic health record data with genomic data and integrating that in some very sophisticated way might be an amazing way to do discovery research, as well as doing studies for genomic medicine implementation and figuring out how things are effective, et cetera, et cetera. This led to something called our eMERGE network, our Electronic Medical Records and Genomics network. that absolutely, and then meanwhile, that it was a pilot in many ways for what's going on now with the Precision Medicine Initiative, otherwise known as the All of Us Research Program, just at a much larger scale. So absolutely, the notion of multidimensional data and bringing in, and coming in from a genomics point of view with these other data, absolutely something of great interest. How you best analyze those data, and here I'll get out of my league quickly, but with machine learning and AI and all the, clearly we're interested. We absolutely have grants in that area, but that also is being looked at a higher level if you just take artificial intelligence as one example, it's being looked at a higher level by many institutes, even at the point of being trans-NIH in nature. There actually is a working group, and this is public, so you can actually, there's anything related to these advisory groups are all very public. You could follow all sorts of meeting reports and things by by finding the right website, Googling it, you'll find it. But Francis Collins, the director of NIH, has his highest level advisory committee, the director. They spin out working groups to deal with different things. They have a working group right now that's working on artificial intelligence in biomedicine. They had an interim report in June. I think they're going to deliver their final report in December. And I almost can guarantee you there'll be funding opportunities coming out of that in some trans-NIH way, maybe in addition to individual institutes as well. So I think all this is absolutely in play and of great interest. And then, it depends. Each institute does it a different way. I'm quite sure, I know for a fact that a big institute like the Cancer Institute, for the huge amounts of cancer data they have probably multiple programs in these data science areas focused on cancer.
This is sort of a bit tangential, but I just thought, because it bothers me a lot. It bothered me when I was at NIH, which is the translational aspect into, not just into product, but into education of the next generation of care providers. That seems that one of the big problems is this is all evolving so quickly. Is how do you evaluate what the findings are, and how do you apply them within these settings, and what is the policy and the infrastructure and the sort of institutional, in academic medical centers, commitment to actually using the findings. I mean, there just seems to be still a big divide and a big time lapse between when those things happen.
There is, and we worry about this at many. I mean, we sort of refer to just the workforce in general. I mean, and you're probably thinking about the sort of a data science lens. I could think of it from a genomic, and particularly genomic medicine lens, of how do we sort of have these practitioners at various levels of the medical system be able to be facile with genomic information. And I mean, there's a huge lag.
Mm-hmm. So let's open this up to questions from all of you relating to the sort of genomic and health related aspects of data science.
So with the Human Genome Project, that was like a really big effort towards a single concerted goal.
Yeah.
So you had a lot more synergy going on. But as a youngster, there's a lot of comparable projects that I look to, like the Encode Project, where they're all very crowdsource driven. So you have all these different investigators that have their own research goals, and they submit data. So my question is, do you think that the state of biomedical sciences now is going to be able to facilitate something that's a large, goal-driven, synergistic thing like the Human Genome Project? Because what I see is there's a lot of challenges with managing data of variable quality and coming from different sources.
Maybe we should try and repeat the question.
Yes. So the question was the observation that the Genome Project was very heavily managed, laser focused, goals very clearly defined. Everybody was sort of at the same goal. And then you gave as an example looking at sort of the set of big known projects now that have different characteristics. You named as an example the Encode Project, which is actually our institute's project, Encyclopedia of DNA Elements, which is sort of our flagship project for finding functional parts of the human genome and characterizing them. And you pointed out how that is different investigators are doing different things, and that maybe the data quality isn't always all the same across it. So the only thing I might say a little differently is you're right that a project like the Encode Project isn't exactly like the Genome Project, because different investigators are contributing different kinds of data that are shedding light on genome function. But keep in mind, they're still quite heavily managed, those projects. We spend our staff spends a lot of time sort of interacting and sort of coordinating the activities of those events. They don't get to do whatever they want by any stretch of the imagination. They're doing it in a highly interactive way, but we want them doing defined things. And there's a number of those projects. There's other projects like the 1,000 Genomes Project, which catalog genomic variants, and there's a Merge Network, which I just mentioned. So, and I think one of your questions was, is that sort of the way these projects are going, or are they going to be more like the Human Genome Project? Was that sort of? Yeah, I think it varies. It's not, we don't run the project, but I mentioned the All of Us Research Program, which is this this national program to enroll 1 million or more Americans to participate in a longitudinal study where their electronic health record data, genomic information will be compiled and shared. That's not quite like the Human Genome Project, because it's really about generating lots of data from lots of different people contributing, but it's a heavily managed project at quite a big scale. So there's an example, something relatively large. I think you're going to see a diversity of types. Even if I look across our institute must fund 15, 20 different consortium. I think it just varies. And then of course, just because you're talking to the institute director whose institute is the most consortium-oriented institute at NIH, you'll still find a lot of other institutes barely have any consortium. I mean, they are mostly about individual investigators getting individual grants to do the things they propose they want to do. So I think it's, but the one bias I will say is I think that team science, interdisciplinary consortium, they're not going anywhere in biomedicine. I'm not saying they're going to take over all of biomedicine, but they're not going to go away. And I think some of the big challenges in biomedical research in the coming decade are going to be, I'm not going to say all of them are going to be tackled, but I think a lot of them are going to really require interdisciplinary teams. I mean, even if you do something as simple as a biologist and a data scientist, but I mean, it's even far more complicated than that. I think it needs a clinician, and it needs a data scientist, and it needs a physicist, and it needs an engineer. I mean, I just think these are the teams that we like to put together. And they're not going away, because I think the challenges are going to require that kind of team.
I will risk putting in a little plug and say, as we develop a new school, we're actually looking at rewarding team science in a way that traditionally doesn't happen in academia.
Completely agree.
But it actually reminded me of something else, which is a little tangential, but it sort of relates to teaming, but not teaming at the scientist level, but at the federal agency level. I mean, I know you've been heavily involved in the idea that the problem is that data is freely shared internationally, but it's actually paid for its collection and its management and everything else, and its analysis is paid for within individual countries typically. And that creates some issues and mismatches. Do you see any progress in that area?
So I'm involved in an initiative at an international level to basically try to look at the international ecosystem of data resources. By the way, ironically, I'll stay away from politics because I could get in trouble if I don't, especially. But I do find it ironic that I was shocked when I became how little interaction there are among international funders around how data resources that all of us depend on every day, whether it's PubMed or whether it's GenBank or it's Uniprot, or I mean, just go right down the line, and yet it's as if everybody just assumes like the sun's going to come up tomorrow. They just assume it'll always be available. And when I became, and I'm responsible for all the model organism databases, and all of a sudden you realize that you could end up modifying things that will affect scientists around the world in unintended ways. The political thing was we actually got a group of funders to meet, and actually we're now starting a coalition. But our first meeting was like 1 month after the last presidential election, and it was when Trump came in office, and I mean, he came out and basically first proposed a massive cut to NIH budgetarily, which ended up not happening. But it was very interesting talking at that first meeting with my colleagues at other funding agencies who literally learned for the first time that, wow, the NIH could be vulnerable, that maybe someday there wouldn't be as much money at the NIH, and therefore we wouldn't be able to support resources that they were completely dependent on. And they just couldn't even imagine a day where PubMed wasn't going to be free to them. So, good news, it hasn't been threatened, but the more important lesson is that the dependencies on these data resources are so significant around the world, including some emerging scientific geographical areas, that we need a better way just to make sure we're better coordinated, and then there's some redundancy and so forth. And so there's a new coalition that of funders coming together to try to better organize things.
Question back here.
Yeah.
You may disagree with the premise of this question, but do you think that qualitative analysis and ethics and legal aspects of this will catch up to the sort of data acquisition and quantitative aspects?
So tell me a little bit more. I want to make sure I
So
understand.
typically, we find out if we can do something, and after that, we start thinking about why and to what extent we should. That's sort of the trend in big data. I think that's the trend in genomics right now.
Yeah.
And I think it really takes people think of it as sort of a zero-sum resource. Either we focus on qualitative aspects or quantitative. And I tend to think of things as both and I'm wondering maybe if there's anything out there that's supporting the other side of things as the technological.
So if I know where you're heading in the question, I guess the immediate thing I'd say is I think genomics. Oh, I'll repeat it. So they say can I just sort of encapsulate some of the societal issues? I mean, some of the societal issues is the science getting ahead of some of the societal issues and consequences of what we're doing? Is that a fair representation? So I think genomics is actually ahead of the curve compared to a lot of other disciplines. One of the things the Human Genome Project did, and and actually Congress requires us to do, is to dedicate a fraction of our research dollars, 5% it turns out to be, to study the ethical, legal, and social implications of genomic advances. So ELSI, ethical, legal, social implications. This ELSI is a phrase you may hear, and it's actually become a brand. I mean, ELSI research, people refer to it, actually is all coming out of NHGRI, all coming out of the US Congress desire to make sure that we were attempting to anticipate, and it is fair to say whether we've been fully successful or not, but I think in many ways we've been ahead of a lot of other areas. So we have a very vigorous ELSI research program and have since the beginning of the Genome Project. If you look at things like the fact that the Supreme Court ruled that you can't patent genes, that was some of the things leading up to that were a series of grants that we funded to try to develop an evidence base to help inform that conversation. If you look at the Genetic Information Non-Discrimination Act, GINA, which is the U.S.'s Congress, a law that has, how many years has it been? 8 years, 10 years, something like that. That was based on a series of studies we did around genetic discrimination that the ELSI research program supported. Now there's a lot of work we have done in the last 10 years looking at how individuals respond to getting back genomic information. And we hope that will help guide decisions about the routines that healthcare professionals will use in delivering genomic information. So I think we have a lot to be proud of. Have we anticipated everything? Oh my gosh, no. No. We have not. But sometimes you can't always. But I think we've been very mindful of that. And but going forward, we know there's continued challenges.
But it's a good question in the sense it makes me think of what we can learn from that we can apply to data science more generally.
Yeah.
Right? And I struggle with that. I don't know. I struggle even with the question because in some ways that data is, at least at some level, when it's human data, is consented in some way. On the other hand, perhaps people give or don't give enough thought to it even there. But what we're doing with a lot of the data that's being used now, which is getting a lot of press around the negative side of data science, of course, is particularly things like social media data, where people sort of give it away, think even far less about what they do with it. So it would seem that we need stronger legislation and more legalese around that. Now, well, it's already overdue in a sense. I don't know how you feel about that, but this is sort of really a serious debate as to, in our minds, and what we're trying to do in the new school, really the future of data science. The negativity that could arise from this could be absolutely huge. And I don't think we have all sorts of ways that we're thinking about this, but in the end, it's really a question of the sense of who owns the data, even in the in terms of one's genome data. It's not clear to me who if you go and have, just say you're part of the All of Us program, there's some exome sequencing done on you, who owns that sequence?
So the All of Us program will say the participant. They're making a position that that's the participant's data.
But you're also agreeing to having it used by others. And then on the other, I guess you're sort of doing that when you click through those boxes on everything we do currently online. But it's, you know, it's clearly totally inadequate.
Yeah.
So I have a question about data sharing. In genomics, you're way out in front. I work at the Center for Diabetes Technology. I'm not a scientist, I'm a lawyer, and one of my jobs is to make sure that the appropriate supply of data is coming in. And so we're not working at the scale of genomics. We're bonding together smaller data sets and trying to achieve the appropriate level of machine learning.
Sure.
And to do that, we rely on NIH data sharing and other sources. But the infrastructure for doing that is really highly involved. And so I just wonder if you have any comments or thoughts. I mean, if there was some pristine database where everything went to at NIH and became a beautiful library, that would make all the lawyers go away. There'd be no more contracts, and we'd have nothing to talk about. Suck the air right out of the room. But is that feasible? How do we see change and how we think about data sharing?
So, so the question basically was recognizing the incredible value of data sharing writ large, not just genomic data, but all data. You know, is NIH sort of developing the routines, the infrastructure, so forth, for being able to fully capitalize on available data so that lawyers could go away. That was sort of what I heard. Yeah, right. Okay. So I would say it's a work in progress. There are some things that are on the horizon that will be coming out for public comments. Probably the best way for me to phrase this, because I can't really give you specifics, but there are policy developments happening, and often when we do policy developments, before we would implement, we would put things out for public comment. So keep your eye out this fall. There will be an opportunity more broadly around data sharing and data sharing plans as part of grant applications, and this is definitely to go beyond genomic data. And so what I would say is, absolutely, this is not just a work in progress, there's a lot of effort being put into this. You know, as somebody who grew up with this ethos of share, growing up within the Genome Project, you know, I fully recognize genomic data is so much easier to talk about and to make rules around and this, that, and the other. And the real challenge when you say something like, well, somebody who gets a grant from NIH, they should just share all the data. You could say that, and then you say, well, what does that really mean? And for anybody who's worked in a laboratory, really, you know, you take a lot of notes in your note. You know, what is real data, and you will just drown in minutia if you would try to get every little piece of data that every little undergraduate and graduate student and postdoc and staff scientist generated. So that's not feasible either. So it's sort of defining it, and then you have to think about what's the format of that data, and you know, again, genomic data is easy. You know, and how do you share it, and how do you organize? So it gets really complicated really quick, and so I think this is going to be a real challenge, and I don't think, nor should it just be the NIH making all these decisions. I think the community, we really are going to have to come to grips with this, balancing the value and the practicality and the expense. I mean, that's the other thing that makes us very nervous is how, you know, we know we're going to have to pay to be able to achieve our value, but you know, we also can't bankrupt the system. So this is still a work in progress, and I encourage you to be involved. I encourage you to, when given an opportunity to provide feedback on requests for information from the NIH, please give feedback. And institutionally give feedback too. That's the other thing. It's valuable to get individual voices, but maybe Phil, this is for you. I mean, you know, when things like that come out, getting groups of people together to come up with an institutional feedback, sometimes that also is very valuable.
Yeah, we've actually in the process of implementing various things that were tried at the NIH that are sort of ongoing. The idea of sort of having open data repositories within the institution. Mm-hmm. And I feel that the library has, in the new world of libraries, that the maintenance and oversight of such resources is part of the mandate of the library. Here with Dean of Libraries, John Unsworth, he's very keen on this. We already have a preservation aspect called Libra, but we don't have a sort of active archive, and we're actually working on that kind of thing. But I don't want to sidetrack into that.
There's another hand out here.
There was one back there.
I'm a physicist, so.
That's okay.
Yes, so I'm here
I just listened to a lawyer. I could certainly listen to a physicist.
I'm here just because I was curious about, so the more I sit here, the more I think about, the way we think about data is clearly, very different. And so for us, the data, there's no question, they have to be out there.
Yeah.
They just okay, let's leave aside there's a classified sector that, especially nuclear physics, we're gonna leave aside. Goes by completely different rules, but that's a small part. Most data have to be accessible because they have to be validated, and they have to be validated down to the small percent. So how do you see? Are we part of this? Are we part of this? Big data. And again, DOE and SF, all our funding agencies, they have precise rules about how we have to handle the data, how we have to manage the data, and so on and so forth. Are we in the same boat as far as data are handled, or are we? What's your vision? Can we just we are all scientists? Okay, that's a common thing. We all have big data. We've had big data. In some sense, the history of physics parallels your history. So we started in the early 90s. It was unbelievable how naive our calculations were, and then things evolved. And so there are very, very similar aspects, but then.
So I think the question relates to sort of a representation of the culture of physics.
The hard sciences.
Hard sciences, right? And my understanding, and again, I'm not young. I actually still don't have the full appreciation of some of my even elder statesmen do about some of these other fields, but I've been told, and I've grown up in genomics and have been told that it is more like the hard sciences in its culture of data sharing compared to the rest of biomedicine. Okay. And so I think, and again, I would welcome other people to weigh in who have their opinions, but I think the culture of biomedical research goes back to a culture of individual investigators working in individual labs in very smallish groups who hold on to their data until they publish it. Even the notion of releasing the data after they publish it is a new concept in some ways, but so I think genomics, is the exception in biomedical research compared to other biomedical subdisciplines. I think that's
your data have to be public or not?
Well, here's what I'd say. The major NIH mandated data sharing policy is in genomics. There's another one that's in clinical trials, so maybe that's the second one. But there is not what's about to go out for public comment and going to be talked about is trying to broaden this beyond genomics. So genomics is so exceptional, and I don't mean that in an arrogant way. I just mean that it's just true. It's just it's so unusual that it actually at NIH has its own data sharing policy because it was easy, because the field is young, and because the field grew up in an ethos like physics. I just don't think the cell biologists feel that way. I just don't think the biochemists feel that way. I don't think, the microbiologists feel that way.
There's a message here. I think you're absolutely right that in the notion of high energy physics and other areas of physics where there's a strong aspect of data sharing. And because first of all, everyone was contributing to the same, the data, large amounts of data were coming off one collider or whatever it is, and many people are using that data. So of course you naturally share that data. That's kind of what happened with the Human Genome Project as well, and then it's gone further into genomics. But if you think about that, and then, as Eric's pointing out, the cultures of other areas within biomedicine, particularly clinical versus basic, where people are not so keen to share. But culture is like one end of the spectrum, and you can start working back. So there's also policies, and then you work back, and then there's beyond. Before you even get to the raw data itself, there's processes, there's software, there's workflows, whatever these things are. And there's the question of and those all have value in different fields. What you can learn from them, and I think one of the things where we're headed, certainly what we're trying to do here, is to create that interdisciplinary sort of set of activities. So when you're doing something, you actually do something. Let's just make this up, in a respective workflow system that's probably completely unknown in biomedicine. it would be good if there was a means of interchanging at least the beginnings of an understanding about what each field might gain from each other, which they don't do currently. And for us, that's what we see as the role of data science. It's really that science of interdisciplinarity. But that's getting a bit off on a soapbox. So sorry. Other questions?
There's a question up here in front.
All right.
This is more from the clinical research side, since I used to work at the NCI doing the cancer research, actually, the clinical cancer research. We're trying to get we also have a similar sharing policy, less so, but a similar one to the Genome projects. However, the infrastructure, if you were to push this out more broadly, would require a large generalized infrastructure. Some of our computers still running on Windows 95, for example, and stuff like that, so they don't talk well with anything made in the last decade. So is there any push internally to kind of bring everything up to a certain level technologically to support the increased sharing of the data science?
Among clinical, so the question is, for clinical data, is there sort of standardization or ways of trying to have more standardized approach? I mean, I think I probably can't cite all the specific examples, but I think the short answer is yes. Some of these are taking place within particular areas like cancer research. Some of them are taking place within big projects like All of Us research program, because they're again trying to harmonize electronic health record data coming from multiple electronic health record systems. And then some of it's just coming more across, clinicaltrials.gov. So again, attempts to sort of harmonize at that level. So I think absolutely the answer is yeah, but it's not taking place in any one pocket. It's probably several.
I did hear that from Patty Brennan, who's the director of National Library of Medicine, that there was a funding call coming that was going to address the kind of thing that you're talking about. So keep an eye out for that.
Yeah.
Other questions? We're almost on the hour. Must be a burning question. Vladek,
We've heard from a physicist and a lawyer. We have to have
at the back of the room.
yeah.
So I'll be the negative Nelly, the other side of the equation. So I'm a biostatistician. And so I'm going to raise the question in data sharing on, I know that we all in research have a goal in NIH with the scientific rigor of the research that is done. But coming from the biostatistics side, I've seen the negative impact of data sharing in terms of the lack of rigor that people may use with data they never collected to understand the subtleties, or should I say, lack of reading of documentation to understand the subtleties. So as NIH is going forward more with the data sharing policy, what sort of things are you guys thinking of in terms of that rigor piece, that data's not enough?
Yeah, well, we appreciate that the metadata is really important in defining what that is. I mean, it's a two-edged sword, right? Because, so I guess I was supposed to repeat the question, which mostly was that, especially through, well, I mean, in general, just because you have data that's shared, it doesn't, for rigor and reproducibility, sometimes if you don't have an adequate amount of information about that data, you could be designing very poor analyses that may be making completely incorrect conclusions based on the findings. So I get that. There's sort of a two-edged sword, because it also seems to me that the more we share data, the more opportunities there are for reproducing findings and shining light where there's a lack of rigor. But on the other hand, it could lead to incorrectly designed or interpreted studies because of a lack of intimacy that you have as if it was your own data. I think it's a fair point. I think it's something that we attempt to try to define the right sets of metadata, but I also think that this needs to be a two-way street. I mean, I think as we want to slowly but surely, share the data, but raise the boat by which we're doing it, increase our survey. So we'll get it wrong at first, but maybe then we'll realize that the kind of we really need these 5 areas of metadata, and maybe those will take a long time to get fully baked in, but it seems to me those feedback loops are going to be needed. But I like anything else, right? Crap in, crap out. I get all that, and so I think we just need to bring sophistication to it, to ask the hard questions at the time we're trying to find out what data needs to be shared.
The problem is we don't know it's crap in until we spend a week trying to figure out what it is, right? So if So I would implore the NIH to start funding the notion of Yelp for data. Okay, so but in a controlled way, so that you could actually read as part of this metadata you're talking about the other people's sweat and toil that went in to try and interpret that data, so that we could save people a lot of time and energy. We're glad it will give you the final word, a brief word.
I think that it's easy to share the data, to get the data, as long as the NIH grant is active. What to do if the NIH grant is closed, and in particular, BD2K created some resources which are used by thousands of people. How they should be funded?
How you mean
Yes,
that?
how they should be funded. Yes, that GM is saying that they will fund resources under condition that they were created from GM money, not from a human genome administrated BD2K. So what will happen?
Yeah, so I don't have an answer. So the answer, the question basically is a combination of questions. One is sort of what happens in the case of grants that are no longer active, and getting data associated with those. The second question had to do with how we deal with the long-term funding of data infrastructures. And what I would say is we don't have that right at NIH, and we're still working on this. I'm struggling. I mean, as you gave an example of GM, I have examples of NHGRI where we're simply historically have been funding things, and whether we could do that long term or not is up in the air. The issue around grants that end and enforcement and so forth, this is part of what we're trying to figure out as we try to expand our data sharing policies. One of the things we want to hear input. I mean, how to do that. And we want to hear from institutions. I mean, at the end of the day, the institutions will have a role to play in this, because while an individual may no longer be at an institution, or they may no longer have an active grant, I mean, the grant was to the institution. And so if there's and that's part of what's going to be that's being looked at now as to what is the expectation for a data management plan for every single grant. And so we need to figure that out. I agree that there'll always be dilemmas about specific scenarios, such as when an investigator is no longer around, but we're going to have to work through those.
So I just thought I have to ask a question, so you don't get the last word. Sorry. Well, so just in one sentence, or if as a new school of data science that's just been formed, from your perspective, from let's just say the genomics perspective, what is one area that one thing that we should be focusing on that could really benefit that community writ large?
The biomedical research community at large. I would say, I'll probably give a few answers. Well, workforce. I mean, that's probably, workforce. There's huge concerns in biomedical research ecosystem that we will not have a sufficient number of people well-versed in data science. So I would say that's sort of a one dimension. I would say another, you wanted me to give a more sophisticated scientific answer, I would say, integration of multidimensional data. Because it's just very clear, as somebody who started with genomic data, when we all of a sudden were able to integrate that with electronic health record data, oh my gosh, it was a whole other world. And we started thinking about integrating that with data that wearable sensors or monitoring environment. I mean, holy cow. I mean, but those are challenges on how to integrate all that and analyze across. So those would be two areas. Keep you guys busy.
So thanks very much, Eric. Join me in thanking Eric for actually wonderful questions.